All right, our next presenter is Himani Patel, and Dr. Turner will introduce her.
Good morning. My name is Ashley Turner. I'm a faculty member of the Department of Biology. It's my pleasure to introduce Himani Patel. She is a new master's biology thesis student coming to JSU. She started at JSU, she's back at JSU now. Her interest is dental health. She'll check your teeth and make sure you flossed and brushed them. But she's going to tell you an interesting story today about using a C. elegans, a nematode model, to study human dental disease. Himani, take it away.
So the disease that I have chosen to study is gingival fibromatosis, and it is a hereditary disease that presents itself as benign overgrowth of gingival tissue in the mouth. So in very severe cases, it can actually encapsulate the entire mouth, as you can see in this image right here, and in less severe cases, it can present itself as a concentrated nodule similar to this. So researchers have found that the SOS1 gene is associated with the gingival fibromatosis disease, and a missense mutation in this disease can, or in the gene, can cause this disease. So what I'm doing is I am finding a missense mutation, variant of uncertain significance in this gene, and determining if it is pathogenic or benign. And if the conserved variant of uncertain significance is identified in the human SOS1 gene and its nematode ortholog, then it will impact SOS gene function and lead to reproductive issues in the mutant nematode compared to the nematode model. So even though the nematode doesn't have teeth, the SOS1 gene in the nematode affects its reproductive health. So if the missense mutation does come out to be pathogenic, then the nematode will probably experience reproductive issues such as being sterile. So the materials and methods that I used Microsoft OneNote to document all of the findings throughout this entire project. And I used the ClinVar database to find variants of uncertain significance in the SOS1 gene. I used benchling to model these genes and visually see and compare the different genes. And Polyphen2 is a tool made by Harvard University, and it predicts if the VUS, or variant of uncertain significance, is pathogenic or benign, and also other genetic and molecular biology agents. So right here I have two graphs, and it shows evolutionary conservation and analysis of the SOS1 gene. So this is at a DNA level, and we're looking at the base pair at the number 3793, and it's changing from a T to a G. And when that change occurs, that is when this disease happens. So on a DNA level, this base pair, it is conserved within all the species that I've chosen. So I chose human, dog, cow, chicken, zebrafish, and nematode, which is the model I am using. And also, it is very conserved on an amino acid level as well. So after doing this, I actually did gel electrophoresis, and right here, this is a picture, a photograph of my gel. And this right here is the ladder, so that's just a marker to help lead the amplification on the gel. And 2-1, this right here, that is my DNA digest of the nematode. 2-2 is Dr. Turner's DNA digest, and 2-3 is the water negative control. So the expected base size, or size of the base pairs, was supposed to be 883, and the amplification actually occurred at 1200, so it is a little higher than what it should have been, or what it was expected to be. And there's also faint amplification in the water, so that should have not happened. I think that could have been issues with contamination or just an issue with micropipetting during PCR. So it would be needed to redo this experiment to troubleshoot any errors that might have occurred. And this is my Polyphen2 prediction for my SOS1 bus. And it actually goes through a scale from 0 to 1, where if it's closer to 0, it's most likely benign, and if it's closer to 1, it's predicted to be pathogenic. So I thought it was going to be pathogenic, just because the bus was highly conserved between all of these different species. But I was surprised that my result came out to be benign. So the score that I got was 0.116, and that's very close to 0. So due to polyphen 2 prediction, this variant would most likely be benign. Future experiments would be needed for me to continue this experiment. So I would choose and select another bus through mutational analysis and polyphen 2 to find one that is pathogenic so I can carry on with this experiment. But I do plan on still analyzing this bus, just because it was a little surprising since it was so highly conserved on an evolutionary level that it still came out to be predicting benign. So both of the bus that I do choose, the future pathogenic one that I will find and this one that I have already found, I will be examining it in silico and in vivo in the meantime. And I would like to give a special thanks to the Department of Biology at JSU for providing support, and also Dr. Turner. Does anyone have any further questions? Yes.
Is this condition treatable?
It is, but it's done through laser, so there's a laser that burns off the gingival tissue, but it does grow back. So it is benign, it's not cancerous, but it does continue to grow back, so they would need to just continuously follow up with those appointments. But you can imagine, that has to be very, very
That
painful.
just seems horrible to me.
Yes.
Yeah.
So there is no permanent solution. Okay. Any more questions?
Well, I think I just missed something. The gel that y'all ran, what was the purpose of running the gel again? What were y'all looking for?
So we were looking at amplification of the DNA strand where that mutation occurred.
Okay,
So
just
we
for
used
that, okay.
PCR to
So
amplify
we're
that.
just looking for that in the wild type?
Yes.
Okay.
More questions? No? All right, let's thank our speaker again.